So, we've really emphasized this idea that we have to remove these amino groups from amino acids, and it has to be excreted as ammonia. The reason for excretion is because ammonia is toxic to animals. We don't entirely know why ammonia is toxic. It's not entirely understood. We do know that ammonia readily crosses the blood-brain barrier. It's toxic to neurons, so it actually alters the formation of synapses and can have general defects in metabolism of cells. If there's really major ammonia intoxication, then it can cause a person to go into a comatose state. It can cause cerebral edemas, as well as increased cranial pressure. So, our bodies have mechanisms for excreting ammonia from the body. So the urea cycle is important for excreting nitrogens from our body. If nitrogens are not used in the synthesis of new amino acids or other nitrogenous products, then those nitrogens in the form of amino groups are then going to be channeled into a single excretory product. These excretory products are really going to be different based on the organism. So that amino group can be excreted as ammonia in fish, or uric acid in birds, or as in urea via the urea cycle. And so in the urea cycle, the net reaction is two ammonium ions will react with carbon dioxide to form urea and water. And the urea cycle occurs in the liver, and some of those reactions are going to occur in the cytosol, whereas some of those reactions are going to occur in the mitochondria. The first reaction that happens, it actually happens before entry into the urea cycle, which is synthesis of carbamoyl phosphate. So carbamoyl phosphate, which is this molecule here, is catalyzed by an enzyme called Carbamoyl Phosphate Synthesis 1, otherwise known as CPS1. It is located in the mitochondria, so this is going to be distinct from CPS2 that's located in the cytosol. CPS2 actually has a role in nucleic acid synthesis, which you'll hear more about in an upcoming lecture. And essentially, in this reaction, the ammonia that's coming from either glutamate or glutamine deamination is going to be incorporated into carbamoyl phosphate. And so this reaction actually uses two ATPs and involves bicarbonate as the carbon source. And so in this reaction, we have bicarbonate will react with ATP, so it's going to be phosphorylated by ATP, forming this carboxyphosphate intermediate. Ammonia will then come in, and it can then attack that carboxyl group and then displace the phosphoryl group to generate a carbamate intermediate. In the next reaction, carbamate will then become phosphorylated by ATP to yield carbamoyl phosphate. Carbamoyl phosphate is really important because it's going to now be an activated carbamoyl donor, right? It's going to be a very reactive molecule for donating this carbamoyl group. So the carbamoyl group is this amino group attached to the carboxylic acid. And so now that we have this carbamoyl phosphate, it can then enter the urea cycle. And so this diagram shows essentially all of the reactions of the urea cycle. Again, here's the net reaction in which two ammoniums plus CO2 will yield urea and water. There are 4 steps of the urea cycle. Some of those steps, again, occur in the mitochondrial matrix. Other steps occur in the cytosol. There are transporters in the membrane transporting ornithine and citrulline, which are intermediates of the urea cycle. It's going to actually use a total of 4 ATPs in the urea cycle. And the amino groups are coming from carbamoyl phosphate. So remember that amino group on the carbamoyl phosphate, as well as aspartate. So we're going to go through each reaction individually. And so in the first reaction, we start with ornithine. So ornithine actually gets transported into the mitochondria matrix. It'll react with the carbamoyl phosphate, where it'll attach that carbamoyl group onto ornithine, forming citrulline, and so an inorganic phosphate will be displaced. This is catalyzed by the enzyme called ornithine transcarbamylase. Now that we have citrulline, the citrulline will be reacted with an aspartic acid to form an arginosuccinate. So the amino group from aspartic acid will be added to citrulline to make our arginosuccinate. So that's where our second amino group is coming in. And so in this reaction mechanism, a rearrangement of citrulline results in attack of the citrulline attacking that alpha phosphate of ATP, resulting in a citral AMP intermediate. Once the citral AMP intermediate is formed, the amino group of aspartic acid will then attack this carbon, which then will displace the AMP, resulting in a covalent bond formed between citrulline and aspartic acid, forming arginosuccinate intermediate. Once we have arginosuccinate will be cleaved to form fumarate and arginine, and then arginine will then be cleaved to form urea and ornithine. Now there is a tight link between the urea cycle and the citric acid cycle. This is called the aspartate arginosuccinate shunt. In this process, oxaloacetate from the citric acid cycle forms aspartate via a transamination reaction that involves glutamate and alpha-ketoglutarate. Aspartate is then transported out of the mitochondria matrix and enters the urea cycle, where it then reacts with citrulline to form arginosuccinate. Arginosuccinate, remember, is cleaved to form fumarate and arginine. That fumarate, which is the byproduct of the urea cycle, is then converted to malate, and malate then is able to enter the mitochondria and re-enter the citric acid cycle, where it's then regenerated into oxaloacetate. Now the importance of this is that this helps to actually generate NADH, which through oxidative phosphorylation will provide 2.5 ATP, which then partially offsets the huge energy investment of the urea cycle. The urea cycle has different mechanisms that can actually regulate the rate by which the real urea cycle is running. And one of the mechanisms is more of a short-term mechanism. So recall that if you have high levels of amino acid catabolism through the transamination reactions, many of the amino groups from amino acids, they're going to be collected on to glutamate. And so high levels of glutamate will react with acetyl-CoA through the enzyme N-acetylglutamate synthase to make this intermediate called N-acetylglutamate. N-acetylglutamate production is then going to allosterically regulate CPS1. So remember, CPS1 is Carbamoyl Phosphate Synthesis 1, and it's that enzyme in the urea cycle that reacts carbonic acid with ammonia to make carbamoyl phosphate. That's that first reaction important for urea synthesis. And so again, the N-acetylglutamate actually allosterically activates that, which then is going to increase this reaction and increase the rate of the urea cycle. So again, you have high levels of glutamate being formed as a result of amino acid catabolism and collection of amino groups, and those amino groups then need to somehow be catabolized by the urea cycle, which the glutamate is then going to stimulate it through N-acetylglutamate. In terms of long-term regulation, you have increased transcription and translation of all of the urea cycle enzymes. This typically tends to happen during starvation or to an individual that's on a high proteins diet. And this is because you have breakdown of, well, at least for those that are undergoing starvation, you have breakdown of muscle protein, which begins to supply much of the organism's metabolic energy. So you would have a bunch of deamination of those amino acids, which is then going to result in amino groups being dealt with to then be removed through urea production. And so you have an increase in the urea cycle to then increase this need to make more urea. Amino acid catabolism of all of the different amino acids are highly complex. Just to give you guys a flavor of some of the reactions that are involved in amino acid metabolism, I'm just going to describe a couple of selected pathways that are involved in amino acid metabolism. And one of these is the mechanisms by which oxaloacetate is formed through the metabolism of various amino acids. Asparagine, through the enzyme asparaginase, can be reacted with water to cleave off that amino group on asparagine to then make aspartate. And then aspartate, through a transamination reaction involving alpha-ketoglutarate, can then be converted into oxaloacetate and glutamate. And so this reaction is catalyzed by aspartate aminotransferase using PLP as a cofactor. And so this transamination reaction is pretty much identical to the transamination reaction that we've talked about previously. The only difference is it uses aspartate as its substrate and produces oxaloacetate. But it still involves alpha-ketoglutarate and glutamate. Serine is an amino acid that can be converted to pyruvate, and this is catalyzed by serine dehydratase. This is an enzyme that also uses PLP in its reaction mechanism. And so the first thing that happens is PLP forms an external aldimine with serine, so they're covalently bonded in the active site of the enzyme. Then there is a base in the enzyme that abstracts a proton, which then breaks the carbon-hydrogen bond between the serine. An electron shift results in the elimination of water, and so water is eliminated. So in the next step, we have reversal of PLP linkage to then form this intermediate, which is an enamine. You then next have a rearrangement reaction that then forms an imine, followed by a hydrolytic cleavage to deaminate the imine, which yields pyruvate as the final product. So phenylalanine and tyrosine can be metabolized to eventually form acetoacetyl-CoA. Therefore, it is a ketogenic amino acid. I know that this schematic is really complicated, but I just want to point out just one reaction that has clinical significance. So phenylalanine can actually be converted to tyrosine, and that is actually converted or is actually catalyzed by an enzyme called phenylalanine hydroxylase. The conversion of phenylalanine to tyrosine is such that it hydroxylate its substrate by adding oxygen, followed by a reduction to make tyrosine. So again, the clinical significance of conversion of phenylalanine to tyrosine by phenylalanine hydroxylase is that if you have a defect in this enzyme, it can lead to PKU disease. So PKU is phenylketonuria, and this is a disease that occurs because you're missing that enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine. And so because of the lack of the enzyme, you actually get buildup of phenylalanine, and then phenylalanine, and as phenylalanine builds up, some of it actually gets converted into an alternate metabolite, which converts phenylalanine to pyruvate. And phenylalanine and pyruvate can also be converted to phenylacetate and phenylactate. And all three of these metabolites are actually toxic metabolites. This disease can actually lead to abnormal brain development and mental retardation. And it's thought that the mechanism by which this happens is because you have so much phenylalanine in the blood, buildup of it in the blood, it actually competes for amino acid transport and is able to cross the blood-brain barrier. And PKU is actually one of these diseases that are actually tested at birth. When newborns are born, they end up getting tested for a variety of genetic diseases, and PKU is tested. And so if a newborn does get detected for having PKU, then in terms of helping the child develop normally, then they just have to follow a strict diet in which they limit their phenylalanine intake and also supplement their diet with tyrosine, and also avoid aspartame, which is a peptide that's found in many different, or a peptide derivative that's found in many different foods, such as diet beverages. So we're going to talk just very briefly in terms of how branched-chain amino acids are metabolized. Branched-chain amino acids such as valine, isoleucine, and leucine, they can be acted upon by a branched-chain aminotransferase, and through a transamination reaction that again involves glutamate and alpha-ketoglutarate, are then converted into their corresponding alpha-keto acids. These alpha-keto acids can be oxidatively decarboxylated and reacted upon to form acyl-CoA derivatives. And so these enzymes are actually really similar to enzymes like pyruvate dehydrogenase and succinate dehydrogenase, which involves coenzymes such as coenzyme A and NAD in their reaction mechanism. It turns out that if there is you could actually have a defect in some of these branched-chained alpha-keto acid dehydrogenase complex, which converts these alpha-keto acids into acyl-CoA derivatives. And so, so if there is a defect in these branched-chain amino acids, then a person can develop what's called maple syrup urine disease, which is characterized by the sweet odor in the urine due to the buildup of these alpha-keto acids, as well as abnormal development and death in infancy if intake of these amino acids are not controlled. And so this table shows some of the different diseases that can occur due to a defect in amino acid metabolism. And we covered just two of those, which is phenylketonuria as well as maple syrup disease. But as you can see, if you have a defect in amino acid metabolism, that is a major deal, and many of these defects are defects that are tested at birth. So we spent a good amount of time talking about amino acid catabolism, and now we're going to shift gears a little bit and consider anabolism of amino acids, that is how amino acids are made. But first I want to go back to this slide to remind us that some of the amino acids, that of the 20 common amino acids in our body, 9 of the amino acids are essential and the rest are non-essential. And they are, so if they're non-essential, that means that our body is able to produce those amino acids. And if they're essential, then that means that we need to consume that amino acid through a dietary source. Now, for the non-essential amino acids, they're going to be coming from metabolic intermediates, such as those metabolic intermediates coming from glycolysis, such as 3-phosphoglycerate that can be used to make serine, intermediates like pyruvate and citric acid cycle intermediates can be used to make alanine, aspartate, and glutamate, and other byproducts of various pathways such as the urea cycle can be used to make arginine. And then also there's what we call semi-essential amino acids, meaning that our body can make the amino acids. They're being made from essential amino acids. They're kind of in between in that they're semi-essential. And that's going to be cysteine, which is made from methionine, tyrosine, which we learn is made from phenylalanine, and then arginine, which can be made from lysine. So all of the amino acids that are the non-essential amino acids, meaning our body can make them, again, they're going to be derived from glycolysis, or they're going to be derived from the citric acid cycle or the pentose phosphate pathway in the urea cycle. And so all of these are different carbon intermediates that can be converted into amino acids. None of these carbon intermediates have an amino group on them, and so nitrogen is going to enter by way of glutamine or glutamate. So glutamine or glutamate are going to provide the amino group for making the amino acid. Now, some of the reactions involved in amino acid metabolism are really quite simple, and some are not. This table again kind of shows some of the precursors that are used to make amino acids. Many of these amino acids are going to have a common metabolic precursor. For example, glutamate, glutamine, proline, and arginine can be derived from alpha-ketoglutarate as a precursor, and 3-phosphoglycerate can be used to make serine, glycine, cysteine, and so on. So many of the amino acids that are produced, they can be produced through a transamination reaction using glutamate or glutamine. This reaction, this transamination reaction, is essentially the same transamination reaction that we saw in amino acid catabolism, except we have, as substrates, we have glutamate and alpha-keto acid, and the products are going to be alpha-ketoglutarate and alpha-amino acid. And so in this reaction, which of course involves PLP and an aminotransferase, in a transamination reaction, a glutamate will be reacted with an alpha-keto acid such that you have a transfer of the amino group onto the alpha-keto acid to make the amino acid, and then this produces alpha-ketoglutarate. For a glutamine transamination that's carried by glutamine aminotransferase, it uses a very similar mechanism, except it does not use pyridoxal phosphate in its mechanism, and instead it uses an active site serine. To give you a taste and an appreciation for amino acid biosynthesis, we're just going to briefly talk about how serine and glycine is produced using a metabolic intermediate. And so in this case, it starts with 3-phosphoglycerate. So remember that 3-phosphoglycerate is coming from glycolysis, and this reaction involves oxidation, transamination, hydrolysis, and carbon transfer. And so in the first reaction, 3-phosphoglycerate is oxidized to 3-phosphohydroxyl pyruvate using NAD+ as the oxidizing reagent. Once we form this amino this intermediate, then we can have a transamination reaction to then convert the ketone group into an amino group, which involves the enzyme phosphoserine aminotransferase using glutamate as the amino donor, producing then alpha-ketoglutarate as one of the products. So now 3-phosphoserine This produces 3-phosphorylserine, and then through a hydrolysis reaction can then cleave that phosphate group off the serine to then produce serine. And then serine, through one additional step by serine hydroxyl methyltransferase, can be converted into glycine, which then is going to remove the hydroxyl group on the serine using PLP, and then serine can be converted into glycine through a serine hydroxyl methyltransferase, which is going to basically remove that the carbon and the hydroxyl group through a carbon transfer reaction to make glycine, and this involves PLP as well as N-methylene hydrofolate as a carbon acceptor. Now, all amino acids are important for our body, and so this is because if we want to synthesize protein, we need all of our amino acids, and if one amino acid is missing or low, then that's going to inhibit protein synthesis. And so some organisms, such as bacteria and fungi and most plants, they have that ability to be able to synthesize all amino acids. However, other species, such as humans, they, in order to get all of the amino acids they need, then they have to acquire some of it through a dietary source. And so recall that amino acids they're kind of stored in, and so one disease that can come about through protein deficiency is called kwashiorkor, which is particularly severe in growing children, and it involves the breakdown of the body's own proteins due to, again, a deficiency in dietary protein. And so the last thing that I want to mention that is probably of more just biological interest to you guys is that amino acids, they are precursors of various neurotransmitters. So, for example, tyrosine can be used to make dopamine, norepinephrine, and epinephrine. Glutamate can be used to make aminobutyrate, also known as GABA. Histidine can be used to make histamine, and tryptophan can be used to make serotonin.